time she'll have to draw me a graph and then take a picture of it and submit it. But uh, we'll get there eventually. So like I said, we're doing this at the beginning of the year because it comes up in every single unit. It's carried across the whole year. Experimental design and setup can be asked along any unit throughout the entirety of the year. So we're going to begin with actually identifying variables. This document should be linked on Canvas, and so you can either just take notes as I take them, or you can print it off, or you can just do it in a journal, whatever works best for you. I'm actually going to start with variables because I think it makes more sense to start with variables and then go back to the hypotheses. So you've talked about and learned about identifying variables before in previous courses. And if not, you're going to have to do some quick learning. But there are 4 types of variables that we talk about. We have our independent variable, which is the variable that you as the scientist set up. So that's the one that we manipulate. So independent, I as the scientist make these decisions. So I say these are the variables that I'm deciding that I'm changing. Then we have our dependent variables. Dependent is data driven. So the dependent is the data that you collect. It's going to be measured or observed. Okay. And it depends on the independent variable. That's why it's called dependent. Then you have control variables. These are used for comparison. These variables are the ones everyone kind of gets confused about. So you can either have a negative control or a positive control. Sometimes you have both. Typically, we always think about the negative one. The positive one is harder because in experimentation, you don't always know what the expected outcome is or the ideal effect. So not every experiment is going to have a positive control, but most of them do always have a negative control or the option to have a negative control. And if we don't have a negative control, think about how we could create that. Okay. So again, a control variable is used in comparison. Scientists compare the experimental group or the group we're testing, the independent variable we're testing, to the control group or the control independent variable to determine whether it was caused by what we're testing or caused by some poor design setup. So a control group just makes sure your experiment is sound and effective. And we'll get to examples about that later. And then you have constants. Sometimes we call these controlled variables, which I know that's kind of confusing with a control group or control variable. That's why I call them constants, but I know in the Bozeman video before this, he called them controlled variables. And these are things that we held constant or controlled throughout the experiment. So just keep that in mind in this course when I'm talking about it. A control variable is a variable used for comparison. It's one of the groups in the independent variable, whereas constant are the factors that stay the same throughout the entirety of the experiment. We have to have constants so that there's no other variables at play that could affect the results we're looking for. So we're going to do this practice, and then you on the practice application that you're submitting this week, you're going to have to do this on your own. So identifying variables. I'm just going to use my highlighters. You can write out the answers. I might write out some of the answers, but with identifying variables, this is really important because, like I said, the AP test will actually ask you to identify the independent, identify the dependent, identify some constants, identify a control group. The AP test will ask you these questions, and most of the time it doesn't ask it as a multiple choice, it asks it as an FRQ. Those are the most straightforward and I would say easiest to get. So those are the ones that you're like, I don't have to remember this random fact about biology. I just have to know how to figure out what variables are which. So those are the points you can get. You can obviously get any of them, but those are the ones that aren't got yous, not that they try to get you a lot, but it's not that random piece of information that you might have forgotten. It's, can you recall what an independent variable is and identify in this situation? So I would say those are the ones that you should lean into learning because it's very doable to get those points. Okay, and so we're going to use this example today in 2 of the 3 practices that we do. So in this experimental design, a scientist wants to determine whether a new fertilizer increases tomato plant growth. Group A receives no fertilizer, Group B receives the standard fertilizer that is known to increase growth, and Group C receives the new fertilizer. All plants receive the same amount of water, sunlight, soil, and are grown for 6 weeks. Plant height is measured every week. So we're going to identify our 4 types of variables, 2 of these. So I guess it's 5. But like I said, I'll either highlight or write up my answers. So for my independent variable is the one that I decide as the scientist. So right here in our setup, right, I decided that I was going to do these 3 groups. So instead of my answer writing group A, group B, and group C, I'm going to have an overall label for it, and I'm going to put types of fertilizer. Okay, so type of fertilizer is my independent variable, right? Because I have no fertilizer, standard, and the new. So the no fertilizer is not really type, but it is a type, or because it's not the amount of fertilizer, right? Because I have a standard and a new one, right? It's just the type or whether or not they get the fertilizer. That would be your independent variable. Your dependent, remember, is the data you collect. So dependent is our data. So what is our result, if you will, not the interpretation, just like what we actually went in and the data we collected. And that would probably be, that would be plant height every week. So our weekly plant height is going to be the dependent variable that is going to depend on the type of fertilizer the plants received. Okay. Then we have our control variables. We have a negative and a positive. For our negative control, this is the group that receives no treatment. So when we talk about control variables or control groups, that's another way to say control groups. I'm going to look at my independent. I'm going to look at my independent treatments and say which of these 3 treatments received no treatment or received none of whatever I'm testing. And in this case, the no treatment would be group A that receives no fertilizer. So my negative control in this instance would be group A. And then for my positive control, like I said, not every experiment is going to have one of these because we don't always know the desired outcome or the expected outcome, right? Right here it says unique controls receive treatment known to produce expected effect, right? In this case, we do have one because I wanted to present it to you, showing you the one that exists. And so it's going to be group B, receives the standard fertilizer already known to increase growth. So when we're testing something new, we might, hey, this is the, leading brand. Let's compare it against the leading brand. This is our positive control. We know this one works. We know it's effective. So we can test the effectiveness of ours against it. Okay. And then we have our constants within the experiment, and you always have to have constants, right? So in this experiment, our constants are going to be amount of water, sunlight, soil. And that's important because say I was changing the amount of water on the different plants in addition to the fertilizer, right? Well, now the growth could be the increase in growth or decrease or lack of growth could be due to the amount of water, not necessarily the type of fertilizer present. So we always have only one independent variable that we're testing, and the rest of the variables around it have to remain constant. And so it's really important to have these aspects of your experiment. Okay, and that's why negative and positive control are so important, because I have no fertilizer. Well, say the no fertilizer group does really, really good when compared. Well, then it makes me think about my constants and makes me think, well, am I keeping everything constant? Maybe my no fertilizer group was, in a spot of perfect sunlight where the other 2 were actually shaded a little bit more. So it helps us remind us, hey, those constants actually truly have to remain constant. Otherwise, the results are invalid. Okay, so that is variables in a nutshell. Like I said, you will get FRQs throughout the year. You'll get some multiple choice, not as typical. Usually it is mainly FRQs that ask these questions, but you'll get some identify these variables. A lot of the times it asks about independent and dependent more than not, but it will also ask about controls as well. I've seen all 4 of these types of variables asked about on AP Bio FRQs. Okay, we're going to backtrack to the previous page, which is hypotheses. So what a hypothesis is a testable statement. When you were in middle school and maybe even early high school, depending on what grade you're in, if you ever taught how to write a hypothesis, they usually start off with that sentence stem, if then. While that is not bad, it is not an invalid sentence stem. It helps you guys to get to thinking, because a hypothesis is a very hard sentence structure to wrap our brain around sometimes. At the end of the day, it's just a testable statement. So it doesn't have to start with if and have a then. That's just what we do early on to get you to kind of thinking about those testable statements. You will have to write both types of hypotheses in this class. And again, I do this because the AP Bio FRQs will ask you to write them sometimes. Not every time. Obviously, I don't write the test, the AP test, so I can't guarantee it, but I more often than not see them at least ask one question on the FRQ, if not two, about writing a hypothesis, as well as some multiple choice questions will reference hypotheses or ask you to identify them as well. So there's two types. One you're more familiar with is this second one right here, which is alternative hypothesis. We put the word alternative in front of it. This is just a normal hypothesis that you usually would think about. So this states a relationship between two variables. So this is related to this, and you need to state how. So whether it has a direct relationship or an indirect relationship, or this causes this to increase, this causes this to decrease, stating some kind of relationship between two variables. There's not necessarily a right and a wrong, right? Obviously, we do the experiment and our hypothesis can be proven incorrect, and that's okay. All a hypothesis is a testable statement, right? So I could hypothesize that sunlight actually decreases plant growth. Sunlight is one of my variables, plant growth is the other, and I'm stating the relationship that it decreases. Now, obviously, we know that that's not true. Sunlight helps increase plant growth overall, depending on other factors, of course, but it's still a valid testable statement, right? I can do an experiment surrounding that hypothesis and either prove my hypothesis correct or disprove it. Okay. And then we have a null hypothesis. These are special in the sense that null, think of no, right? So in a null hypothesis, it's pretty much always stating the same thing, which is that there's no statistical relationship or no difference between two variables. And so when we talk about statistics later next week, we'll talk about some wording with that. But the null hypothesis states no relationship. So this is going to be pretty much the same for every experiment that we do, in the sense that you'll look at the two variables we're, comparing or exploring the relationship, and your null is always going to be that there's no relationship between A and B, right? Whereas your alternative, it could have a plethora of ideas. A and B are direct, A and B are indirect, So those are the two hypotheses that you will be writing every time you do an experiment. You'll have to write both of them because it gets you to thinking, gets you used to writing them. That way, when you have to, if you have to on the AP test, you're prepared for that. So with that being said, we're going to stick with the same example from last time. So we identified those variables last time, right? We had our dependent variable, which was plant height. I'm going to do it in a different color so you can actually read it. Plant height was our dependent variable, and our independent variable were these groups right here, or types of fertilizer. So I'm going to start with the alternative because I feel like that one helps us write our null. And so for alternative, we're stating the relationship between the type of fertilizer and plant height. So we can say the type of, sorry, I sometimes write in cursive, sometimes print, and sometimes all over the place. The type of fertilizer does affect, and that might be the wrong effect, but that's okay, does affect plant height. Now, this is kind of very vague. I personally like it to be a little bit more specific of the type of relationship, right? Obviously, I'm just saying that it affects it, but I want you to go a step further and tell me how it affects it. So if we were to write it, what I would deem is, a better, I'm going to erase that a little bit because that says that they have a relationship. So the type of fertilizer, no, I don't write it like that. See, you're getting, you're my brain. So we're going to word this in a way to where we're a little bit more specific, right? So the new fertilizer will have an increase in plant height over 6 weeks. This is a bit more specific, and the reason why I stress this is because in this class, when you're doing labs, I want you to be specific. I don't necessarily just want you to tell me plant height, and type of fertilizer is related. That's the bare minimum. If you're on an IP test, FRQ, and you're like, I don't know what to do that, okay? But in this class, if you want the full points, I'm trying to get you to write it more specifically because it's more correct. So I like this one better. A null is going to be, again, not as specific just because it's typically always the same. If you can remember null, no relationship, you can write these every time. So again, we're comparing the relationship between the type of fertilizer and plant height. So I would write the type of fertilizer has no relationship or no relation to plant height. Or I could say does not affect plant height, is not related to plant height at all. You can word it a bunch of ways, but it's saying the same thing, okay? Boop. So that is hypotheses. In a nutshell, again, we'll get used to and we'll practice rating those throughout the course of the year. The last and arguably the most important piece of this lesson is graphing. So you've graphed before, I hope. And so graphing shows us or displays data that's collected. And then we get to interpret that data based on the graph. You will be looking at so many tables and graphs this year, and you will be drawing so many tables and graphs this year. And that's good. It's important. It helps you analyze data, and that's such a real world application, regardless of the AP test, regardless of anything else. Like, being able to look at data and actually factually interpret it is so important in today's society because you can also see when people are doing it incorrectly and you can see when things are not valid data. But most graphs have 5 main parts: a title, a x-axis, a y-axis, or independent and dependent variable, a scale or labels on the axis, and a legend. Okay, there's the definitions right there. I'll tell you right now, for the AP course, the most important thing is going to be the independent dependent variable, scale, and then I'm also going to write and axis labels. Boop. And that's really important for the AP test, so important. And I'll get to that. It all has to do with the FRQs, but those are the most important thing. Titles are important, I would say, in life as a scientist, not so much for the AP test specifically, but that's just good to have on anything. So I always appreciate a title. And then the legend or key are important. Not every graph requires one. Okay. Again, here are the definitions. You can read those on your own, go through those on your own. I'm assuming you understand what those are because this is an AP level course, and in a college level course, you would have known what those were already, right? We're in a college level course right now. Rules for graphing. So always use a pencil typically. However, on the AP exam, you're gonna use a pen. And I'm here to prepare you for the AP exam. So typically in like an actual college course, if you were to turn in a graph or something, you would use Excel or Google Sheets. It'll graph it for you. It's more accurate, right? Because it's not being graphed by hand. On the AP exam, they don't let you do that. You have to graph by hand. So that's the skill we're going to really practice throughout the year because I want you to get all the points you can because it is on an FRQ. And so because of that, we're going to work on graphing in pen because when you do it for the AP test, you have to do it in blue or black pen. There is no pencil. You are not allowed to use a pencil on that exam. And so I really want you to get used to graphing in a pen because a lot of the times we make mistakes when graphing. And so the more we practice it in pen, the more we can at least set ourselves up for success in that regard, okay? So when you turn in a graph for me, it's typically going to be done in pen rather than pencil because while I appreciate the pencil, I understand we all make mistakes. On the AP test, you don't get to, erase it and start over. You have to scratch it out, move over, and improvise, if you will. Okay, your independent variable is always on the x-axis. Your dependent is on the y. There's that acronym dry mix. And to be honest with you, I just know this way back from the day when I was in school. Dependent y-axis, independent x-axis. I think this stands for manipulate, and this stands for results, maybe? I don't really remember what the R and the M stand for, but dependent y, independent x. Okay. Include all the parts. You need a title. axis labels with units. That is not the same thing as your scale. Your scale is going to be the actual plot points. You need a legend if it requires multiple colors. And then you're going to need multiple, or if you have multiple subjects use different colors or pattern lines on the AP test, you're going to have to use different pattern lines because you have a blue or a black pen. If you use blue and black, they can't tell the difference in the color because what it happens is they photocopy your FRQ. That's why it has to be in blue or black pen, so they can't tell the difference. It just looks like dark ink. And then you have to choose an appropriate labeled graph. Typically, we only see the first two. I don't think I've ever seen a pie chart or a pie graph. But line is going to be measuring the change in something over time typically, or if your x-axis is in numbers, I'll explain what that means here in a minute. Your bar is comparing individuals to each other with only one data point. So if your x-axis is words, so think if like I polled everybody and I said, what's your favorite color? My x-axis would be things like red, orange, yellow, right? Those are the words that I'm talking about in your scale. So this practice problem is a little different. So experiment 1, use the following data to create an appropriate graph and answer the questions. Graph paper on the next page. It's not on the next page, it's right here. So here is my table. Whenever you're graphing, you will either collect the data and make your own table, or you'll be given a table. On the AP test, you are given a table. Okay, a little shortcut to help you out is the first column is typically your x-axis, and the second column is typically your y-axis. Now, as we can see, we have a third column. So when I really look at the data, let's see what we're actually working with here. We have depth in meters, and so that's there. We have bubbles per minute for plant A and bubbles per minute in plant B. So obviously we submerged these plants, I suppose, in water, I'm assuming. And so those went to the depth. And then at each depth, we found how many bubbles were produced per minute per plant. So this is actually going to be also our y, because we just have two different scenarios. So this is going to be one where you're graphing multiple subjects. So we're going to use different colors or patterned lines here. Because this is not an AP level problem, excuse me, hiccup. I'm just going to use multiple colors. So here we have our blank graph. If this was just a normal lab, I would put a title. For the AP test, you don't actually get any points for titles, so I wouldn't worry about that. You do get points for labeling your axes, though. Okay. So we're all good at copying here, I'm sure. That's the joke I used to always tell my students, right? So for your labels, just copy the labels they give you here, if applicable, right? Which it's here. So my X label would be depth in meters. So I would just write depth, and I'm going to put that in meters. And then my y-axis is bubbles per minute. Okay, and that's all I need. You don't need to put plant A or plant B because you're going to write a legend here in a minute. This one, the y-axis doesn't really need a label. A unit per se, because the unit is bubbles per minute. Or you could write bubbles and then in parentheses per minute. I guess that's would be your unit. Okay, so now we're going to determine our scale. For your scale, you don't have to write a number at each individual line. That'll take you forever. As long as you're counting by the same number every time, and as long as they're spaced out by the same distance, you're good. I typically say do at least 5 numbers, okay, for a line graph. I forgot to mention that. This is going to be a line graph. How do we know it's a line graph? Because our x-axis is numbers right here. Notice these numbers do not count by the same every time. That's okay. Again, for your scale, as long as they are counting by the same number and have the same distance, you will put everything on there. So what I would do is I would count all these lines. So let's go ahead and count them real quick. So it's about 31. Okay, so I'm just going to say 30, right? And we go to 30 right here. So what I would do is I'm going to start with 0. I know my smallest number is 2 and my smallest number is 2. My largest number is 30, so I at least have to go to 30. I'm not going to write 1 through 30 at the bottom. That would take me forever. So I'm probably just going to count by, we'll do by 5s. Okay. I didn't even need to do that much personally. Like I said, as long as you have about 5 numbers here, what I tell my students graphing throughout the year, if you're graphing by hand and you don't have graph paper, if you just make a little line like that and you did, 0, 30, divide that in half, 15, divide that in half, 7.5. What would this be? 23? No, 22.5. That would be enough. Okay. And so we did that. We're done there. We did our x-axis label and x-axis scale. It's correct scale because it's divided evenly and it counts by the same number every time. Notice it is not necessarily these numbers. These numbers are included here, right? Included 0 to 30 is there, 0 to 30 is here. I'm just not going to necessarily put them on those lines, and that's okay. Now I need to make my y-axis scale. So I'm going to look for my smallest number, which appears to be 11. Yeah, 11, I believe. And then I'm going to look for my biggest number, which I believe is 51. Yes. Okay. I'm going to count how many lines I have here. If you don't have lines, you can just divide it evenly. You're eyeballing it, and that's okay. I think it's about 34. I think I'm just gonna kind of eyeball it, to be honest with you, and divide half. And that's perfectly fine. On the AP test, typically they give you a kind of graphed piece of paper, but sometimes it's literally just those 2 lines. So again, the lowest was 11, the highest was 51. So I'm just gonna go to 60. So 0 to 60, about halfway is right here. So I'll say that's 30. About halfway in between that is 15, and about halfway between that is 45. I'm done. Like, please do not waste time on the AP test, nor on any of your tests in here, writing each individual number. That is a waste of time. But do notice how I used the entirety of the graph. Always recommend it. Okay. So now I'm going to actually graph. So with that being said, I'm going to have plant A and plant B. So at a depth of 2 meters, it's about 29. So 2's right here. I'm going just under 30, which I said was right there. So maybe that's right there. Then I have 36 at 5. Okay. 36, probably about there. So I'm going to keep graphing. I want you to graph. You can pause or skip ahead. I'm going to do this real time though. So just take a second to graph it yourself. I'll be graphing mine. Okay, so I did plant A. You don't have to connect the dots. You can. I personally don't think you should. And the reason why is because if you connect the dots, that's saying you took data at each individual point, means I took data at 0 meters, 1 meter, 2 meters, 3 meters. So I just leave them blank. For my little key or my little scale, I'll draw a key right here. And so I know that my blue is, so I just leave them blank. For my little key or my little scale, I'll draw a key right here. And so I know that my blue is plant A. Okay, let's choose a different color for plant B. I'll choose a brighter one and I'll make it this red plant B. And so then I'll plot plant B. Give me a second to do that. I'll give you a second to do that. Okay, so I've planted, I plotted point B or plant B now. And so this would be my final graph right here. You can also write a title. Like I said, to me, titles aren't as important because the AP test doesn't count towards them. But if I were to write a title, a good title has both variables listed. So it would be, let's say we have our depth in meters and our bubbles per minute on each type of plant. So let's see, bubbles per minute based on plant depth and type. So based on the type of plant and the type of depth. There's my title right there. So I listed both variables in there, and then we did it across multiple types of plants. And so that's how you would graph it. And I can see the trend, right, as I got right, we peak, I guess, interpretation around the depth of, what is this, 16 meters. Plant B did the best, whereas around 10 meters, plant A did the best. Okay, and then we have to write our variables. We already kind of identified that there. So I'm going to let you write them in, or you can just, draw an arrow. There's your independent. There's your dependent. What could you add as a control group to this experiment? So I want you to think about that. Think about negatives and positives. And so kind of go to your thoughts in there and write your own answer for that one. Okay. And so that's the notes. The next thing you're going to do is the practice doing all of these things together, right? Identifying variables, graphing, and writing hypotheses.